Okay, so welcome to this next video in the playlist on the cardiovascular system. Okay, right. We are looking at the endothelial-derived relaxation factor. So far we've seen that if we give an intravenous infusion of acetylcholine, or an intravenous injection of acetylcholine, it's going to act on the M3 muscarinic acetylcholine receptors on the endothelial cells, and it's going to cause IP3 to go up everywhere within the endothelial cell. Now, let's look at what IP3 going up globally within the endothelial cell is going to do. Well, it's basically going to produce us a calcium signal within the endothelial cell, which is going to take the form of a calcium wave, basically. So, if we look at the sarcoplasmic, well, sorry, not the sarcoplasmic reticulum, the endoplasmic reticulum, because we're not working in the muscle cell anymore. If we look at the endoplasmic reticulum, the endoplasmic reticulum basically stretches across the length of the entire cell, like so. Okay, right. And basically, along the length of this endoplasmic reticulum, which I'll draw again out here, so let's copy that picture down here and do it slightly bigger. Okay, now in the membrane of the endoplasmic reticulum you have what are known as IP3 receptors. Okay, so I'll draw one of these here. And these are effectively calcium channels. Okay, so you have loads of these IP3 receptors all the way along the membrane of the endoplasmic reticulum. So let's have another one here. Okay, right. Now, the calcium level within the cytoplasm of the cell is very, very low, usually kept at around 100 nanomolar. So the concentration of calcium intracellularly is generally around 100 nanomolar. Okay? Whereas concentration of calcium in the endoplasmic reticulum here is very, very high. So this is the endoplasmic reticulum, which I'll just denote as ER. Okay, so if we open calcium channels in the membrane of the endoplasmic reticulum, what you're going to get is calcium level inside the cell going up, basically. Okay, so let's have a look at what IP3 is going to do to the IP3 receptor. And for this, we need to have a look a little bit more in detail at the structure of the IP3 receptor. Okay, so if I draw a big IP3 receptor out over here. Okay. So, basically, the IP3 receptor consists of 4 subunits. So here we go, 4 subunits. It's made of 4 different proteins, okay? And they all come together to make the overall IP3 receptor. Now, when you're making an IP3 receptor, there are 3 different genes which all encode for proteins which can be used as a quarter of the IP3 receptor. So if you want to make an IP3 receptor, you can either make a homotetramer, which is basically when you use the same gene to code for 4 proteins. You make 4 proteins which are all identical, and then you stick them together, basically. That's a homotetramer because you've used the same subunit in all 4 slots, basically. Or you can make heterotetramers. So heterotetrameric IP3 receptors do exist, where basically the different subunits of the IP3 receptor are encoded for by different genes, basically. Okay, last fun fact about the IP3 receptor is that each one of these subunits that makes up the IP3 receptor, and I'll highlight one here, so we'll highlight it in blue. So each one of these subunits that makes up the IP3 receptor here, it is massive. It's a massive great protein. It's around 2,750 amino acids. So when people write alpha, that means amino acids, okay? So it's around 2,750 amino acids long. Okay, so it's a massive great protein, and you put 4 of those together to make the IP3 receptor, often just denoted as the IP3R. Okay, so this overall IP3 receptor is a massive great protein complex. Right. Now let's discuss what IP3 is actually going to do to the IP3 receptor. So for this, let's have a look at this top view of the IP3 receptor. And remember, this is just a cartoon rather than an actual representation of the crystal structure. Okay, so here is my cartoon top view of the IP3 receptor. So we are looking effectively at the cytosolic domain of the IP3 receptor. Okay, so the theory is this, that before IP3 has bound to the IP3 receptor, then every single one of these subunits of the IP3 receptor has what is known as an inhibitory calcium binding site. Whoops. So these are all inhibitory calcium binding sites, these orange things. So this is an inhibitory calcium binding site. Inhibitory calcium binding site. So there are 4 subunits making up the IP3 receptor. So there are therefore 4 inhibitory calcium binding sites. Okay. Now, each IP3 receptor also has an IP3 binding site. Okay, here. Now, the theory is this, that before IP3 binds to its IP3 binding site, shown in pink, the IP3 receptor has this inhibitory calcium binding site exposed, and if calcium now binds to that inhibitory calcium binding site, it's going to decrease the probability that the channel is open even further. Now, the probability that the channel will actually be open at any given time is very low anyway at the moment when IP3 is not bound. But if calcium binds to these inhibitory calcium binding sites, it will reduce it even further, basically. Okay, right. Now, the thoughts, what the speculated mechanism by which IP3 activates these receptors is that when IP3 binds to the IP3 binding site, and of course there are 4 IP3 binding sites because you have 4 subunits, so you're going to need an IP3 molecule to bind to each one of the 4 subunits, basically. So you're going to need an IP3 to bind to the binding site of each one of the 4 IP3 subunits. So overall you're going to need 4 IP3 molecules to bind to the overall IP3 receptor. Okay, now when IP3 binds to the IP3 receptor, what's believed to happen is it changes the conformation of all of these 4 subunits. And I shouldn't have drawn this pore larger, I do apologise for that, that's misleading. It hasn't caused it to open yet, so ignore the pore being larger, that's really bad. Okay, what it does cause is it causes the inhibitory calcium binding site to be removed, basically. It's no longer exposed, and instead what happens is you expose a new calcium binding site, which is a stimulatory calcium binding site. So this binding site in green is a stimulatory calcium binding site. So IP3 binding does not cause the receptor to open. Instead, it removes the inhibitory calcium binding site and it exposes a stimulatory calcium binding site. Okay, now if you want the IP3 receptors to open, all you need to do is bind calcium into these 4 stimulatory calcium binding sites, basically. Okay, so what's going to happen then? When IP3 goes up in the entire cytoplasm of this endothelial cell, it means that IP3 receptors all the way along this endoplasmic reticulum are going to bind IP3. So this one's going to bind IP3, this one's going to bind IP3, this one's going to bind IP3. All of them are going to be bound to IP3. That means they're all going to change conformation to retract their inhibitory calcium binding site and expose their stimulatory calcium binding site. So this is a stimulatory calcium binding site. Okay, right. Now all that needs to happen is one of them needs to get calcium to bind to all 4 of its calcium binding sites. If just one of them achieves that, then all of them are going to achieve it, and I'll show you how in a moment. So, the calcium concentration in the cytoplasm, I said, was very low, approximately 100 nanomolar. However, it's not 0. So there is a chance that one of these IP3 receptors will just happen to get 4 calcium ions binding to it, i.e. 1 in each of the stimulatory calcium binding sites on each of the IP3 receptor subunits. That will cause this IP3 receptor to open. So let's say it was this one. It will then open. Now when it opens, it's going to allow calcium to leave it, basically. So calcium is going to come out, and calcium now is going to rise in the vicinity of this IP3 receptor. So calcium is going to go up nearby this IP3 receptor, okay? Okay, now what's going to happen is it's going to spill over onto its neighbours. So calcium will spill over from this single IP3 receptor onto all of its neighbours, and obviously I've drawn 3 here. There will be millions of these things in the length of the endoplasmic reticulum. So it's going to spill over to its nearby IP3 receptors. Now all of those have got IP3 bound to them and are therefore in this primed state, just waiting to receive 4 calcium ions bound to them. Now that this calcium has gone up so much, they're going to get 4 calcium ions binding to them, and therefore they're going to open. So this one here is now going to open, and that means that calcium is going to leave through it into the cytoplasm as well. So you're going to get calcium going up in the vicinity of this one. Okay. So what has happened is this one opening here has caused all of its neighbours basically to open. Now, when these neighbours open, the calcium level nearby them is going to elevate, and that's going to spill over to their neighbours, so this one over here. And then this one again has got IP3 bound to it and primed, so when calcium arrives there, it's going to cause this one to open and release calcium. So basically you get this propagating calcium signal that's going to propagate along the cell, basically. Now, that's propagating calcium signal is what is known as a calcium wave. Okay, but I haven't told you the full story. That's not it finished yet. The calcium wave is more complicated than this. So let me show you what the calcium dynamics is going to look like by basically saying, let's call this point 1. Okay, so this is point 1. Let's say this is point 2 here. And let's say this is point 3 over here. Okay, and they're sort of like, they're meant to be actually in the cell, and I've drawn them outside the cell, but we're measuring calcium from around here, we're measuring calcium from around here, and we're measuring calcium from around there. So let's draw a graph of what it's going to look like, basically. Well, how am I going to do this? Because I don't want to lose that picture. Where can I draw this? I can't draw on there, though, so I'm going to have to get a new piece of paper. So I'm going to get a new piece of paper, but I'll keep referring to this picture. So let's say we draw a graph, basically of calcium concentration, so this is calcium level here, versus time. Now, if we draw it for position 1 of our cell here, then initially what's going to happen is this IP3 receptor is going to open and calcium concentration is going to go up, basically. So what we will see is something that looks like this. However, you might now expect that it would just remain high, because the IP3 receptor is just going to remain open, the calcium is going to remain in the cytoplasm, you know, you would expect it to remain there, but I haven't told you the full story. What's going to happen is at low calcium concentrations, the calcium will bind to these stimulatory calcium binding sites that are now exposed on the IP3 receptor, and they will activate it to open. However, when calcium concentration around this IP3 receptor gets too high, it stops having a stimulatory effect on that IP3 receptor and instead starts having a very much so inhibitory effect, a negative feedback. So when calcium gets too high, it starts negatively feeding back on this IP3 receptor and inhibiting it. So these IP3 receptors are going to close in this region after calcium gets to a certain level. So let's say here. So they've closed now. Okay, so what's going to happen to the calcium now? Well, there are pumps in the endoplasmic reticulum membrane, known as the, I'll write this down here. Okay, so this pump in the endoplasmic reticulum membrane, which I'll draw again here. So this is the endoplasmic reticulum membrane. Here is this pump. This pump is known as the sarco/endoplasmic reticulum. endoplasmic reticulum calcium ATPase. Okay, and its role is to return the calcium from the cytoplasm into the lumen of the sarco or endoplasmic reticulum. Okay, and because sarco/endoplasmic reticulum calcium ATPase is a bit of a mouthful, we often abbreviate this to SERCA, sarco/endoplasmic reticulum calcium ATPase, SERCA. Right, and basically what SERCA does is it moves 2 calcium ions, so here are 2 calcium ions, back into the ER lumen in exchange for free protons moving back into the cytoplasm. So free protons are moved out of the ER lumen into the cytoplasm in exchange for 2 calciums going into the ER lumen. So let me give SERCA a colour. So here is this pump in green. Now, in order to achieve this feat, it also needs to hydrolyze ATP. So it's going to take ATP basically to ADP. So here's ATP to ADP, adenosine diphosphate, and an inorganic phosphate molecule. So you're going to get ADP being produced and also an inorganic phosphate. So it has to hydrolyze ATP in order to get the energy needed to perform this movement. Okay, right. So this SERCA pump is in the membrane of the endoplasmic reticulum, and you have loads of them, just like you have loads of IP3 receptors. So, once calcium gets to a certain level, it's going to start inhibiting this IP3 receptor. So the IP3 receptors in this vicinity have now closed, okay? Now what's going to happen is SERCA is going to start pumping all the calcium back in, okay? Right, so what that means is that calcium is going to go back down. Okay, right, so that's what the calcium level for position 1 looks like. Now let's look at the calcium level at position 2 over here. Now, basically position 2, calcium will be low initially, and then when the calcium spills over from position 1, basically, it will cause the IP3 receptors at position 2 to open and release calcium from the ER, and then cause calcium levels at position 2 to go up. So you would expect position 2's calcium levels to rise slightly later than position 1's, and then what will happen is again calcium at position 2 will get so high that it will start inhibiting the IP3 receptors, and then the SERCA pump will return it into the endoplasmic reticulum. Okay, right. And finally, if we if we look at position 3, position 3 is going to have its calcium level spike in response to position 2's calcium spiking. So calcium will overflow from position 2 onto position 3, activate the IP3 receptors, and calcium will be released from the ER. Okay, so you'll get the calcium spike for position 3 even later still. So really, a calcium wave is this propagating spike in calcium level. Now, there's one final thing I need to tell you. This is not just calcium wave. Well, this is one calcium wave. So this is a calcium wave at the moment. Basically, when you stimulate an endothelial cell and you raise IP3 levels in the cytoplasm, you don't just get one calcium wave. You get repeated calcium waves, basically. So one of these will occur. The calcium spike will propagate from position 1 along the endothelial cell. Okay, it will end, and then it will do it all over again. So if we look at this graph, what will happen is later on, you'll get the whole thing starting again. So if this is the graph position 1, calcium will go up, so this is position 1 again. So let me highlight them in a certain colour to denote what position you're at. So position 1 will denote in blue. So position 1 goes down to around 100 nanomolar and then spikes back up again. So it undergoes another calcium wave, basically. And then what will happen is the same thing will happen again. It will propagate forward. So again, you'll have position 2 undergoing the calcium spike slightly later, and then finally position 3 undergoing it later still, like so. Okay, so let me highlight position 2 and position 3 as well. Right, so we'll do position 2 in red, like so. And this is going to get a mess if if I highlight over each other. Okay, there's position 2, and then finally we'll have position 3 in orange. Okay, right. So, basically you get repeated calcium waves, and there's this interval basically between consecutive calcium waves. So this interval here between consecutive calcium spikes at a particular point, this is dependent upon the IP3 concentration. Dependent upon IP3 concentration. So if you have a higher IP3 concentration, which indicates that you have a higher concentration of acetylcholine stimulating the production of the IP3, you get a shorter interval between calcium spikes, i.e. you fire calcium spikes more frequently. You fire calcium waves at a higher frequency. Okay, so the frequency of the calcium waves propagating across these endothelial cells is dependent upon the IP3 concentration, which is dependent upon the stimulus intensity of the acetylcholine. So a higher stimulus intensity, a higher frequency of calcium waves. A lower stimulus intensity, a lower frequency of calcium waves. And I want to emphasize that if I put more acetylcholine onto a cell, I get higher IP3 levels, but I don't get a higher calcium spike at position 1. So what does not happen is you do not get bigger spikes the higher IP3 concentration you have. Instead, you get more frequent waves, basically. A calcium wave is a calcium wave. I would compare it, basically, and a very good comparison for it is action potentials occurring across neurons. When you want to, basically an action potential is an all or nothing principle. You either have one or you don't. You don't have differing degrees of action potential. Okay? So a neuron can fire an action potential or it doesn't. And if you want to encode like the stimulus intensity, maybe how much pressure you're putting onto a mechanoreceptor, that is not encoded by how larger amplitude this action potential is, because an action potential is an action potential. The amplitude of the electrical potential difference change does not change depending on how high intensity the stimulus is. Instead, what you have to do is you have to encode it by the frequency of action potential. So if you're pressing on harder, that's going to trigger more frequent action potentials. Similarly here, the size of these calcium oscillations at each region do not change depending on the stimulus intensity. Instead, the frequency of these calcium waves is what changes depending on stimulus intensity. Okay, so now we've discussed the calcium signaling, the calcium dynamics in quite some detail. In the next video, what we'll do is we'll discuss what the calcium is actually going to do within the cell.